Alright, we have a lot to talk about, so let's get right to it. What you probably know about DNA is that it's the genetic material that we are all made of. But what does that mean? Where did the concept come from, and how is it discovered? All of these questions will soon be answered, but first let's go over a basic understanding of DNA. First off, DNA is organized into a chromosome. It doesn't just sit loose in the nucleus of a cell. DNA is wrapped around proteins, called a histoprotein, and characteristically makes the shape of an X. Its left side is a mirrored copy of the right. Up until the early 1950s, no one knew what DNA did. The structure of DNA is very simple, made of a sugar, a phosphate, and a nitrogen-containing base. Scientists assume that the genetic material of a cell must be very complex and varied because genetics seemed very complicated. Only one biomolecule seemed complex enough to be the code for all of life, a protein. Today we know this is not true. In fact, we have all the knowledge now about DNA that we didn't before. Let's start off by talking about the people that are responsible for our knowledge of DNA. Alright, the year was 1928, and medical doctor Frederick Griffith was looking at bacteria to try to figure out which were causing diseases. To further his research, he tested mice to find out more about genetic materials and if he could create a hypothesis on the matter. The photo here shows an experiment that Frederick did on 4 different mice. He used the drug Streptococcus pneumoniae in each of the mice, but in different ways. The first experiment he tested, which is actually the second picture, is a mouse that was injected with rough Streptococcus pneumoniae, which means that when it was growing in a tray, it appeared rough. This mouse survived the rough strain. It was healthy and was able to live. The second experiment that Griffith performed is shown in picture number 1. Griffith wanted to determine the pathogenicity of his smooth strain of Streptococcus. Pathogenicity is the disease-causing capability of a bacteria. Griffith injected the living cultures of his smooth strain into the mice. 2 weeks after injection, Griffith found that the mice were killed as a result of the introduction of the smooth bacteria into their systems. The smooth strain of bacteria causes disease in the mice and therefore is pathogenic. Griffith wanted to determine whether the viability of the smooth strain was required for the pathogenicity. To do this, he first needed to kill the bacteria by boiling them for a short period of time. He wanted to determine whether the factor present in the living smooth bacteria that caused disease could be transformed to non-pathogenic material. Griffith first boiled pathogenic smooth bacteria and needed to mix this heat-killed smooth bacteria with living rough bacteria. After mixing the 2 strains, Griffith injected a mouse with the mixture. Neither the rough bacteria nor the heat-killed smooth bacteria were capable of causing disease on their own. The mixture was injected into mice, and the mice were incubated for 2 weeks. After 2 weeks, Griffith found that the mice died. He proposed that when the smooth bacterial culture with heat-killed components was present, it caused the bacteria to be pathogenic. When non-pathogenic rough bacteria was introduced into the culture, the cellular components from the smooth bacteria were able to enter the living rough bacterial cells. Once inside, the cellular components then transformed the living rough bacteria cells into living smooth cells. Griffith therefore determined the cellular components transforming factors. It took 30 years to determine that these transforming factors would one day be known as DNA. Those responsible for this discovery were Avery, McCarty, and McLeod. They wanted to find out what the transforming factor did. They reviewed Griffith's experiment to try and find out the transforming factor. They wanted to know what happened between the transfer of the heated, smooth strain to the rough strain. They did this by breaking down the bacteria. In this breakdown, they had RNA, proteins, and DNA. They used the enzymes which were to break down each of these to see which could transfer the bacteria again. They tested both RNA and proteins and found that they could transform. But after adding a deoxyribonuclease, which breaks down DNA, they found that it was the only one that could not transform. Avery, McCarty, and McLeod were the first to discover that DNA was in fact the transforming factor. Even with this great discovery, most of their work was disregarded as people couldn't believe that DNA was the genetic material that surrounded all living things. Even then, people were still arguing whether or not DNA or proteins were the genetic makeup of all living things. Hershey and Chase designed an experiment which was also known as the Blender experiment that used bacterial phages, which were viruses designed to infect bacteria. A phage infects the bacteria and then creates more of the kind that it wants. Hershey and Chase set up one experiment using sulfur, which is found in proteins, to infect the bacterial atoms, and then observed the color that came out. The second experiment, they used phosphorus found in DNA to infect the atoms. In conclusion, the DNA was a hereditary material because they found evidence that it was the only transforming factor. Moving on in history, the first known scientist to study the structure of DNA was Erwin Chargaff. He looked at organisms to see the different ratios of guanine, thymine, cytosine, and adenine. He saw that the amounts of A and T were the same, and the amounts of G and C were the same. A and G being purines, which were 50% of DNA, and T and C being pyrimidines, which were also 50% of DNA. This was called Chargaff's rule, the knowledge that A and T were equal and C and G were equal. Now, Chargaff didn't know what to do with this information, but two men by the names of Francis Crick and James Watson did. Maurice Wilkins took pictures of crystallized materials. Rosalind Franklin didn't get along well with him, but incidentally they had the best data together. They looked at DNA trying to figure out its structure. James Watson sat in on one of Franklin's secret meetings, which later helped him to figure out the structure of DNA. Crick and Watson knew the structure of a helix coming from the work of Wilson. They knew about the A, G, C, and T, which Erwin earlier discovered. They used models to figure out the structure. The model Watson and Crick worked with had A and T equal and G and C equal. It was a double helix. They are given credit for the discovery and share the Nobel Prize with Wilkins. Unfortunately, Rosalind Franklin died before she could receive the award. Alright, now last but not least is the Michelson-Stahl experiment. After Watson and Crick made sense of Wilkins and Franklin's work and were able to come up with the shape of DNA, the double helix, the only unanswered question thus far was how it made copies of itself. There were 3 different scenarios that Stahl and Michelson had to deal with. The first was semi-conservative scenario, which was that the DNA unfolded in the middle and copied new templates on either side, and Watson and Crick proposed this. And then there was the conservative theory, where DNA wraps around a histoprotein and then duplicates by splitting in the middle, making new templates on either side. The third case was just the dispersive model, which was that DNA would clump into a nucleotide segment and each of these would be copied. So to prove which theory was right, Michelson and Stahl grew E. coli in heavy nitrogen. Heavy nitrogen means that there's an extra neutron, so it's 15N is how you would write it. And so they spun that in an ultracentrifuge, and any DNA containing heavy nitrogen would float to the bottom of the tube. And so they bred a bunch of atoms of heavy nitrogen, and they switched them to live in an E. coli environment that had normal nitrogen. They then observed what happened, and in conclusion they found that this hybrid concurs with hypothesis number 1. And there you have it. We've learned about how DNA was first discovered, how its structure has been studied and observed over the years, and finally how we know that DNA is truly the genetic makeup of our lives. Before I go, I just want to let you know that the reason that many of the early experiments were performed on animals and other means rather than on chemicals is simply lack of materials. Scientists in the early 1900s and up until more recent times did not have proper technology and the same high-tech equipment that we have today. Today, purified chemicals are used in experiments, and as animals' kidneys and other organs were physically designed to break bacteria, using them as testers wouldn't make results accurate. Thank you so much for listening, and I hope that this helped.